Hey everyone, welcome back. So today we are taking a super quick break from our heavy next generation sequencing pipeline for a rapid fire Python pro tip. Okay, so this is a trick you can use right now in your VS Code notebooks to make your scripts run faster and look way more professional. So way back in the previous video, I taught you how to count the nucleotides in a DNA string, right? But the way I taught you was the beginner way. So today I'm going to show you the pro way. So let's jump straight into the notebook. Okay, so let's generate a slightly larger DNA sequence to test this on. Okay, so first of all, we will import random. Okay, after that, let us create a random DNA sequence. Okay, so basis will be uh, A, right? Then T, then C, and then G. Okay, and let's just make a DNA sequence of about 1 million bases. Okay, so we will name it long DNA is equal to uh, dot join random dot choices and basis k is equal to uh, 6 zeros. Okay, that should be sufficient, and then we will print that we have generated uh, length of long DNA uh, base pairs. Okay, let's run this and okay, so we have our sequence. So now if you want to know how many A, T, C and G's are in this sequence, the standard beginner approach is to use the count method for separate items, right? So previously, how did we used to do it was count uh, A is equal to long DNA dot count A, right? And similarly for other sequences also we did the same thing, if you might remember, like this, right? And finally we will print something like uh, A would be something, B would be something, C and G would be something. Okay, so I'll just copy it from the previous code, right? That is how we used to do it previously. If we run it now, it will print something like this. Okay, this works perfectly fine, but mathematically it is highly inefficient. Why? Because you are forcing Python to read that entire 1 million letter string 4 different times. Once for A, then for G, then for C, and then for T. So if you're doing this on a whole genome, it will take forever. So what is the professional way? So professional Python coders do not do this. They use a built-in module called collections, specifically a tool called counter. So let's see how it works. Okay, so we will import from collections import counter. Okay, okay, that library is there. So next we will do base counts is equal to counter and long DNA, and what we have to print is basically base counts, and let's see what happens. Look at that. In exactly one line of code, counter reads the string from left to right exactly once, keeps a running tally of every unique character it sees, and spits out a dictionary-like object with the totals. It is drastically faster, it uses less computer power, and it takes up less space in your script as well. But wait, there is one more thing. Because base counts acts like a dictionary, you can extract the numbers for just G by typing something like this. Okay, like base counts G, some base counts and G. If you run this, you will see the exact value of G basis, right? But counter also has a secret superpower, like more secret superpowers. What if you want to know the most common base in the sequence, right? So for that, we will do most abundant is equal to base counts dot most common. Yeah, that's the command. And 1. Okay, this will find the single most common nucleotide. Okay, so we will print this as well. What we will print is the most common base is, and here we will print most abundant. Let's print and see what happens. And see, A is the single most common nucleotide. So the most common function instantly sorts your counts from highest to lowest. If you pass it the number 1, it gives you the top result. Okay, if you were analyzing a sequence with weird characters or like ends, this instantly tells you the dominant base. And that's your pro tip for the day. So stop using count multiple times in a row if you are handling extremely large sequences. Okay, and instead of using it multiple times, you can use collections.Counters that we learned today. Okay, it is the ultimate tool for generating frequency tables in Python, whether you are counting DNA letters, amino acids, or even words in a clinical text file. So I hope this quick tip saves you some time and processing power. In our next video, we will be heading back to our Linux terminal, okay, and proceed further. So drop a like if you learned something new today and subscribe for more bioinformatics contents. I'll see you in the next video.